Good afternoon, and thank you all for attending today's Journal Club webinar sponsored by the ATS Assembly on Respiratory Structure and Function. My name is Niyati Borkar, and I'm a research fellow at the Mayo Clinic. I'll be co-moderating today's discussion with Jason, entitled Cell Population Resolved Multiomics Analysis of the Developing Lung. Today's webinar will include a presentation by Mereena George Ushakumary, who is a biomedical researcher and scientist at the Pacific Northwest National Laboratory, affiliated with the Biological Sciences Division, where she conducts research in lung biology, cell proteomics, developmental and injury responses in pulmonary system. She has authored and co-authored multiple research articles in areas such as fibroblast biology, cell signaling, lung development and injury, as well as phosphoproteomics published in peer-reviewed journals. Um, Marina's work and her recent publication on cell population resolved multiomics atlas of the developing lung was recently published in the American Journal of Respiratory Cell and Molecular Biology this last year, where she does a deep dive into the molecular profiling of each distinct lung cell type across the lifespan using both proteomics and transcriptomics. We are so excited to learn from you, Marina. A special introduction of our expert panelists who have taken the time to join us today. First, we have with us Dr. Naftali Kaminski, who is a Boehringer-Ingelheim Endowed Professor of Internal Medicine and the Chief of Pulmonary Critical Care and Sleep Medicine at Yale School of Medicine. Building on prior leadership at the University of Pittsburgh, Dr. Kaminski has become a globally recognized pioneer in genomic and systems biology approaches to chronic lung disease. His groundbreaking work includes some of the earliest applications of large scale transcript profiling and high throughput genomics in IPF, COPD, asthma, and sarcoidosis. He has generated extensive single cell data sets and human lung cell atlases, including a recent article published in Nature Communications just this past week. Alongside research, Dr. Kaminski is also deeply committed to training the next generation of physician scientists, mentoring numerous MDs and PhDs who have gone on to establish successful independently funded careers. His leadership extends to service within the ATS, and his contributions to the field have been recognized with major honors, including the ERS Gold Medal for ILD in 2016 and the ATS Amberson Lecture Award in 2022. Thank you so much, Dr. Kaminski, for joining us today for this webinar, and we are looking forward to learn from you as well. Over to you, Jason.
Hello, I am Jason Gokey. I am an assistant professor at Vanderbilt, and I work on lung disease and lung repair, injury repair, and mostly in the epithelium. And I today get to introduce one of our expert panelists, Dr. Anne-Karina Perl, who is a professor in the Division of Neonatology and Pulmonary Biology at Cincinnati Children's Hospital and the University of Cincinnati College of Medicine. Her research focuses primarily on molecular and cellular mechanisms associated with lung development, lung regeneration, and chronic lung diseases. With a primary focus and being a major pioneer in understanding the role of fibroblasts and mesenchymal cell biology and crosstalk, as well as extracellular matrix biology and remodeling. And Dr. Perl has made foundational contributions to defining lung cell identities in the developmental programs using multiple genetic models, including lineage tracing, integrative molecular approaches, and she's also a founder of the International Lung Fibroblast Task Force dedicated to standardizing nomenclature and classifications across the field, which we all enjoy our cell names, and she's helping to clarify that across the field. I can say from personal experience that Anna also is an excellent mentor to the next generation, is really supportive of young scientists. And for recognition for her contributions to respiratory science, Anna received the 2025 ATS Distinguished Achievement Award from the American Thoracic Society last year.